Regarding the urine production, we have three major steps in the urine formation. It's going to be filtration, tubular reabsorption, and tubular secretion. The first one is going to be from the capillaries to the tubules. We're going to filter plasma, and then we're going to get the filtrate in the tubules. It's going to be through a passive movement, osmosis. It's going to be passively. The filtration doesn't need energy. Then we have the second process. It's going to be the tubular reabsorption. Most of it is going to be in the proximal convoluted tubule, and maybe it's going to be 65% of it. And it's going to be through osmosis for the water. And we have some elements. They're going to be reabsorbed passively by facilitated diffusion, and some other by symport or active transport. And we have the tubular secretion, and it's going to be from the capillaries actively through the to the tubules. And maybe this is going to be for substances that were not filtrated here, such as the hydrogen ions or maybe the metabolites of drugs. This second image is going to be about the effective filtration pressure. At the end, we're going to use 10 millimeters of mercury in order to get the plasma in this direction filtrated by the membrane. It's going to be here in this direction. Then the flow is going to be from the capillaries to the Bowman capsule. Regarding the transport in the reabsorption, actually, for the solutes in the proximal convoluted tubule, we have the microvilli here, and then we need to cross in order to get the fluid and solutes from the tubule to the capillaries. We need to cross the cell and then the membrane of the capillaries, the layer of cells. And we have several kinds of transports. The most important one is going to be the osmosis for the water. Remember, here is going to be maybe 65% of the reabsorption. Then we have for glucose, for potassium, and amino acids, we have facilitated diffusion. And remember, these two don't require energy. Then we have symport. It's going to be a protein transporting on the same way, in the same direction, two or more molecules. In this case, it's going to be particularly for potassium and chlorine. And finally, we have the active transport for sodium. The next segment for reabsorption is going to be the loop of Henle. And remember, we have the descending limb and the ascending limb. In the descending limb, we have permeability for the water. We have osmosis for the water. And we are going to be reabsorbing water from here, from the filtrate to the capillaries. And we have some diffusion from the capillaries to the tubule about solids. Then we have the only impermeable tubule section, which is going to be for water, and it's going to be the ascending limb of the loop of Henle, and it's really not permeable at all for water. And on the opposite direction, we have the possibility to diffuse the solids. Then the only piece without permeability for water is going to be this section. The next process is going to be the secretion, and in this particular case, it's going to be the secretion of the hydrogen ion and potassium into the renal tubule. Remember, it's going to be actively and specific from the capillaries to the renal tubule. We have for the hydrogen ions, we have the transport going to be antiport because they're going to be exchanged for sodium. And regarding the potassium, we're going to have secreted into the filtrate by antiport as well. in the distal convoluted tubule. This last process to form urine is very important in order to eliminate the metabolites of drugs, and it's very important to regulate the pH because we're going to be able to eliminate the hydrogen ion through the tubule, and we're going to be able to keep the bicarbonate and sodium ions imported across the basal membrane into the interstitial fluid, and then they're going to be diffusing into the peritubular capillaries. Regarding the urine concentration mechanism, we have three of them. We have the countercurrent mechanism, we have the medullary concentration gradient, and we have the hormonal mechanism. The fluid is separate in structures flowing in opposite directions. That's the meaning of countercurrent. We have the liquid or fluid going into opposite directions. We have descending limb of the loop of Henle, but we have the flow of the blood vessel, the peritubular is going to be opposite, and it's going to be the same on the ascending limb for the loop of Henle. Then we're going to have the peritubular blood going down. Other important mechanism in order to concentrate the urine is going to be the medullary concentration gradient, and as you can know, the interstitial fluid is going to be around 300 millismol per kilo with the same of plasma. This is in cortex, but this is going to be in medulla. At the bottom of the medulla, we have maybe 4 times the osmolarity. It's going to be more dense, and this is another mechanism in order to concentrate the urine. The other mechanism is going to be the production of antidiuretic hormone and the renin mechanism. The antidiuretic hormone is going to move water by osmosis from the less concentrated filtrate into the more concentrated interstitial fluid. Other mechanisms, hormonal mechanisms, they include the renin, angiotensin, aldosterone production, and the renin is going to be produced by the juxtaglomerular apparatus and the nephron. Then we're going to be able to convert the angiotensinogen in angiotensin 1 and 2. The second one is going to be a potent an incredible vasoconstrictor. Then the angiotensin 2 is going to stimulate the production of aldosterone. If you remember, that's going to be in a hormone, a mineralocorticoid, and which is going to be able to transform the concentration of sodium and potassium. Actually, as you can see here, the aldosterone is going to be, the main effect is going to be over the distal convoluted tubule, and it's going to be increasing the sodium reabsorption and the potassium and hydrogen secretion. If we have hypertension, if we have high blood volume and high elevated pressure, then we're going to have several mechanisms in order to get back to the normality. It's going to be homeostatic mechanism. One of them is going to be decreasing the ADH, the antidiuretic hormone production, in order to decrease the water reabsorption. And then if we have less water in our blood vessels, we're going to have less blood volume and less blood pressure. The other mechanism is going to be through the mineralocorticoid aldosterone. Because we have less aldosterone, we're going to have more atrial natriuretic hormone is going to be produced by the heart, and then we're going to be losing more sodium and water through the urine. And finally, we have the baroreceptors in the blood vessels, the sympathetic division, and when they detect high blood pressure, the consequence is going to be increasing the renal blood flow and the rate of filtrate formation, and then we are able to lose more water in our urine. On the contrary, if we have low volume or low pressure, then we're going to have the sympathetic division by receptors. They're going to be increasing the decreasing, sorry, the renal blood flow, and then it's going to be less urine produced. We're going to have less HCl natriuretic hormone, and then we're going to have more aldosterone, and we're going to get more sodium and more water into the blood, increasing the volume and blood pressure. And finally, we're going to have the production of the antidiuretic hormone in order to save more water inside the body. Here we have three important concepts specifically for the depuration of any foreign substance, maybe a drug, maybe some foreign chemicals, and that is the plasma clearance, which is the volume of plasma, the amount of plasma that is clear of a specific substance each minute. The ideal substance is going to be the inulin, and we have two more concepts, important concepts. The tubular load is going to be the total amount of substance that passes through the filtration membrane into the nephrons each minute, and this should be less than the tubular maximum, because tubular maximum is the maximum rate at which a substance can be actively absorbed. This is important, for example, in patients with diabetes. For example, when we have a patient with normal glucose, maybe 100 milligrams per milliliter, then our load is really behind the tubular maximum. That's the reason we have no glucose in our urine. But when you have a load surpassing the tubular maximum, such as 320 milligrams per minute, then you're going to have glucose in your urine. That's the reason patients with diabetes, they have glucose in their urine. The uterus and the urinary bladder, they have four layers. They have the transitional epithelium, which is going to be the innermost layer in contact with urine. Then we have the connective tissue, which is the lamina propria. Then we have the third layer, which is the muscle. Specifically for the bladder, is going to be the detrusor muscle. And finally, we have this nutritious and protective layer, which is with blood vessels, the outermost layer, which is the adventitia.